ground beneath our feet contains a little-known ecosystem harboring abundant life. Research has revealed an incredible web of life. Even plants and microbes converse with each other. Can we harness this web of life for commercial uses? And researchers have found the Prince Charming who wakened sleeping beauties in the soil. This time on Science Zero, we explore the hidden web of life in the soil.
Hello.
Hello.
This is Science Zero.
Our subject this time is soil organisms.
It's an astonishing ecosystem. You almost wonder who could have designed the soil beneath our feet. Today's commentator is philosopher Masao Kurosaki.
With urban sprawl, there's so much concrete, we hardly come in contact with the soil anymore. When I was a kid, playing in the dirt, but now we've left that world behind.
There's no dirt.
True, you never see any around town, but a microcosm within the soil, that sounds intriguing.
Well, let's get started. In this tube, I have some soil from a forest, about 1 gram. Kurosaki-san, about how many living things do you think there are in this 1 gram of soil?
I guess around 100 or 1,000.
Actually, according to a 2002 study by Norwegian researchers, there are almost 5 billion individual organisms in this soil.
5 billion?
Yes, and that's just the number of bacteria.
The human population on Earth is like 5 or 6 billion. That little bit of soil contains as many organisms as there are people on Earth?
Yes, in just 1 gram.
That's astonishing.
What kinds of organisms live in the soil? I went to peek inside the world of soil.
Yasu-san visited Yokohama National University.
Surrounded by trees like this, I don't even feel like I'm on a college campus. I'm here to observe soil organisms. A professor here is going to be my guide, but I don't see him.
Yasu-san!
Yes? You must be Professor Kaneko.
Yasu-san's guide is Nobuhiro Kaneko. He's a soil biologist who studies soil ecosystems. The campus has a grove of trees that were planted 30 years ago. Inside the grove, there is an ideal place to observe soil organisms.
Oh, a big hole.
Yes, we excavated it for teaching purposes. I'll show you. You can see the soil structure.
The hole is 120 centimeters deep, showing what lies beneath the surface. Yasu-san goes into the hole to see.
I see there are layers of different colors.
That's right. The black color shows that this soil is rich in organic matter, such as decomposed leaves.
In the layer of black soil, the top 30 centimeters is especially rich in life. What organisms live near the surface?
Take some of this soil.
Okay.
Do you see the little pellets?
Yes, lots of little pellets.
Do you know what those pellets are?
They're soil, right?
Yes, of course.
But what shaped it like this?
It doesn't happen by itself.
Ow! Sorry.
Yasu-san reflexively threw away the soil. Why? Because she hates worms, and one poked its head out of the soil. These pellets are actually excreted by worms.
Worm casts are formed year after year and remain in the soil.
I see.
Here's a small one.
Okay, wait. Sorry. No, I can't. It's too gross.
When I pour this liquid over the surface, something unusual will happen.
Kaneko-san demonstrates a remarkable phenomenon. He begins to pour a liquid over the surface. It is water with mustard dissolved in it. A minute later,
They're coming out.
Oh, wow.
worms poke their heads out of the soil. So do woodlice, millipedes, and other organisms. The stinging liquid drives them out one after another. Many such organisms live concealed in the soil. But not all of them are large enough to be seen with the naked eye.
Wow, look at them all. Very colourful.
The red creatures are a kind of springtail, about 1 millimetre in length. The small white ones are mites. About 1,000 organisms on this scale occupy an area of soil the size of a footprint. And that's not all. Now let's look under a more powerful microscope.
I see glowing dots.
These little dots that glimmer like stars are stained bacteria. The largest are about 10 microns in size. The bacteria come in many kinds, including types of bacillus and actinomycetes, known for producing antibiotics. The soil also contains molds and other fungi, including Aspergillus oryzae and Penicillium, familiar due to their use in making food and medicine. But the microbes that can be identified make up only a small fraction of those existing in the soil. Most species remain unknown.
Today you showed me all kinds of soil organisms. I was amazed at how many there are.
The microcosm of the soil contains many tiny organisms, too small to see with the naked eye, that scientists are still investigating. So in a sense, although we don't know what's out in space, the soil of our own planet is the final frontier for biology research. That's what I think.
It's almost creepy how many things are living in the soil. So many species.
And now let's introduce a man who can tell us more about things that live in the soil. Associate Professor Kazuhiko Narisawa of Ibaraki University is a specialist in molds that live in the soil.
How did you get involved in research on soil molds?
Well, my reason was that I love to eat tasty foods. Well, that may sound irrelevant.
No, go on.
Growing delicious foods depends on soil microbes. Once I learned that, I had to start studying soil microbes for the sake of the delicious foods they make possible.
That's why?
We heard that most organisms in the soil remain unknown. Why haven't they been discovered yet?
Well, traditionally, research on microbes, such as the ones living in soil, required culturing the microbes to figure out precisely what they do. But, actually, more than 99% of the microbes living in the soil cannot be cultured in the lab.
And why is that?
One of the reasons is that scientists don't quite yet fully understand what nutrients the microbes need to consume in order to grow. Another reason is that the microbes don't live independently. They are interdependent with many other species in the ecosystem. We've recently realized that trying to isolate and culture one microbe species is a mistaken approach.
A certain key word is cracking the secrets of unknown soil organisms. Here it is. Endophyte.
Endophyte? What's that?
Well, the endo in endophyte means inside. The phyte means plant. Combine the two roots and it means organisms that live inside a plant. Most endophyte species are microbes. It would be ideal to know absolutely everything happening in the soil. But there is no way human intelligence could figure all of it out. Most of what goes on in the soil remains a mystery. However, many endophytes can be found in the tissues of plants. And among those, the area around the roots is where many make their home. By limiting our inquiry to a certain area, we can figure out what's happening.
One endophyte that has long been known is in here. This is a soy plant, and look closely at its roots. Do you see the little round things? These are called root nodules. Now, inside the root nodules, we have these endophytes called nodule bacteria. The black dots inside these root nodule cells are all nodule bacteria. And researchers have captured this symbiosis being initiated. Here we have nodule bacteria around a root. The bacteria are the moving things. This photo shows the moment bacteria enter the root, the bacteria appear as green dots. As one approaches a root hair, the tip wraps around the bacterium. The bacteria that have been wrapped up multiply inside the roots, growing into these thread-like structures. When they reach the root, they invade the plant cells and multiply there, causing nodules to form.
So how is this kind of symbiotic relationship established?
On the plant side, a certain chemical is exuded. The bacteria respond to the chemical and establish symbiosis. But this only occurs in legumes. This relationship is exclusively between legumes and nodule bacteria.
So nodule bacteria won't do this with other plants?
No, they won't.
Amazing. In fact, besides the legumes we just saw, other plants also have ingenious symbiotic relationships with microbes. In fact, one type of endophyte is found in 80% of terrestrial plants. It's called mycorrhizal fungus. I'm sorry, I know it's a bit of a mouthful. The latest research has uncovered the details of how symbiosis between plants and mycorrhizal fungi is established.
We're at Osaka Prefecture University. Koki Akiyama studies the exchange of chemicals between mycorrhizal fungi and plants. He is researching a typical mycorrhizal fungus called arbuscular mycorrhiza, or AM fungi. It's a symbiote of around 80% of terrestrial plants. AM fungi extends its fungal filaments into the cells of plants. Then it forms a tissue called an arbuscule. The AM fungus uses the arbuscule to supply the plant with phosphorus from the soil, which plants need to grow. In return, the plant supplies sugars that it produces by photosynthesis. For a long time, it was a mystery how this symbiosis is initiated. Previous research suggested that the plant initiated the symbiosis. Akiyama-san tried to isolate the symbiosis-initiating signaling chemicals from the solution in which the plant's roots were immersed. Signaling chemicals are usually unstable molecules present in minute quantities. No one had ever succeeded in isolating them. By shortening the process to less than 10 minutes, Akiyama-san successfully isolated the chemical.
This is strigalactone.
The chemical he isolated is called strigalactone. How do mycorrhizal fungi respond when a plant exudes this initiating chemical? Akiyama-san observed AM fungi in the presence of strigalactone. This Petri dish contains AM fungi spores and paper soaked with strigalactone. After a while, the tips of the filaments branch out. Once the strigalactone secreted by the roots reaches the AM fungi, the fungi quickly branch out their filaments in order to increase the probability of reaching the root. Also, in February 2010, a German research team published a fascinating report. At the upper right of the screen, the tip of a fungal filament is approaching a root. Now the root glows green. This indicates that the plant genes related to symbiosis are being expressed. When do the genes switch on? Let's watch again. The root is already glowing. But notice that the filament has not yet touched the root. A signaling chemical is being secreted by the AM fungi, prompting the plant to prepare for symbiosis.
Plants secrete strigalactone, which alerts the AM fungus to their presence. It says, "I'm here." Then the AM fungus responds by secreting a molecule that says, "I am an AM fungus," and that's how the symbiosis is initiated. I think that understanding this conversation by means of chemicals is a fascinating area of research.
I understand that something amazing was happening there, but is it really significant that the plant glows green before directly touching the fungi?
Oh, very significant. The plant responded without physical contact, meaning that some chemical is being exuded by the fungi.
Ah.
This first step must take place for symbiosis to occur. It selects the symbiotic partner. How is the partner selected? Through the intermediation of these chemicals. That's what this discovery means.
Researchers have also learned that this molecule, strigalactone, has other functions besides attracting AM fungi.
Here we have a happy little fellow down in the soil. He is an AM fungal spore. When strigalactone is secreted from the plant's roots, the AM fungal spore reacts by germinating. The strigalactone actually exhibits another function in the above-ground part of the plant. It promotes branching.
So one molecule is involved with both AM fungi and above-ground growth. Why is that?
Well, when plants are colonized by the AM fungi, it means that they are now able to obtain needed nutrients from the surrounding soil. For plants, this means their situation is now secure, and they can start to grow. So, the same chemical in the above-ground part of the plant acts as a plant hormone to spur growth. It is basically saying, get bigger, we're ready to grow.
These are ingenious symbiotic relationships with nodule bacteria and mycorrhizal fungi, but there are still more endophytes.
Yes, there are. This is Suzuki grass. The parts you see glowing blue are a bacterial endophyte living inside the Suzuki grass. It's an endophyte called Clostridium. It is believed to play a role in nitrogen fixing. Now, this is a photo from my own laboratory. You can see some black stuff around the edge there. This is also an endophyte, a fungal endophyte. It's called Phyalocephala fortinii, another type of endophyte.
What does this endophyte do?
This endophyte also supplies nitrogen to the plant to support the plant's growth. One last image. The structure in the center that looks like a sac is a plant cell. And snaking upward through the middle of the plant from below, penetrating the plant from outside, is a kind of filament. I think you can see it. This is the fungal filament of an endophyte. Normally, if a microbe invaded a plant's tissue like this, the plant would respond in some way to repel it. But this plant is not reacting at all, as this photo gives proof. The endophyte and the plant have a very friendly relationship.
So the plant is okay with this one coming in. If it were some hostile organism, the plant would shut it out.
Yes. Yes, to shut it out, the plant would build up the cells surrounding the intrusion. But, as you can see here, there has been no cell buildup.
About how many endophyte species are believed to exist?
We have no idea. I would say that it's impossible to even estimate. But there seem to be many such relationships in nature.
So far, we've seen that lots and lots of bacteria live in the soil. But many of these bacteria exist in a dormant state, until something comes along that wakes them up and they start multiplying. Researchers long suspected that dormant bacteria were sleeping beauties waiting for a prince, the Sleeping Beauty hypothesis. But who is the prince? Kaneko-san and his team at Yokohama National University set out to find out. What did Kaneko-san identify as the prince?
Here it is.
What? A millipede? The train millipede is known for eating soil. Is Prince Charming really a millipede? Kaneko-san took identical soil samples and added millipedes to one and not to the other. After a month, he analyzed the DNA content of the two samples. He confirmed that the two were different. The horizontal axis indicates bacteria type, and the vertical axis the amount of each bacteria type. There are more peaks in the graph for the soil that had millipedes. The number of bacteria species jumped dramatically. Does millipede Prince Charming kiss the bacterial Sleeping Beauty? No, he gobbles her up. Right along with the soil he is consuming. The millipede's stomach is both moist and full of nutrients, so the bacteria rapidly multiply.
In fact, the same results were obtained with worms too. Kaneko-san believes that all soil-eating animals have this effect. Now the question for Kaneko-san is what these bacteria that become active in the millipede or worm gut actually do.
When worms and other such creatures eat soil, they leave behind casts in the soil, as Yasu-san saw earlier. Those casts then become a haven for bacteria and other microbes.
It really drives home the point that all life, even down to its smallest forms, is connected in an interdependent web.
Yes. Previously, researchers were focused on isolating individual microbe species to investigate their properties. Now, that's all changed. And the mainstay of research is studying the various interrelationships.
And we have an amazing image of such a mutual relationship to show you. Here it is. This photograph was taken last year by researchers at Ibaraki University.
This is an amazing photo.
Yes, it is.
These vertical tube-like structures you see are fungal filaments. Inside the fungal filaments there are clusters of green dots. These are actually bacteria that are living inside the fungus. Fungi are considered aerobic organisms, which means they need oxygen to grow. However, some species of bacteria are anaerobic. They can live without oxygen. Seeing that amazing photo through the microscope made us think that a fungus with anaerobic bacteria living inside it might be able to grow under anaerobic conditions. So we did an experiment. And the fungus did indeed grow extremely well. So, how do the bacteria benefit? Next, we turn to antibiotic compounds to which bacteria are vulnerable. Bacteria cannot grow when subjected to antibiotics, but fungi are unaffected.
Antibiotics have no effect on fungi.
No effect. So we experimented to see whether antibiotics would work against the bacteria living inside the fungi in that photograph, and they didn't work.
The fungi protected them.
Protected the bacteria.
So in exchange for enabling the fungus to survive without oxygen, the fungus keeps the bacteria safe from harm.
And there may even be more to the relationship.
Narisawa-san is carrying out research to harness endophytes for agriculture.
This is soil that we took from a forest on the island of Yakushima.
He treks all over Japan and occasionally abroad to collect soil samples in a quest for unknown endophytes. So far, he has isolated around 2,000 strains, which he keeps in a freezer. Of these, only 10 or so are known species. Most of the endophytes remain unnamed, their properties unknown. Narisawa-san is interested in the disease-preventing effects of endophytes. He has been experimenting on asparagus, a vegetable whose cultivation in Japan is rapidly expanding. When repeatedly cropped on the same plot, asparagus becomes susceptible to pathogenic soil bacteria. To combat this, Narisawa-san turned to an endophyte he collected in Canada. He planted asparagus shoots with and without the endophyte in soil harboring the pathogen and compared the results after 1 month. On the right is an asparagus plant with the endophyte, on the left one without. The difference in the branches is obvious. The difference is even starker in the roots. The plant without the endophyte sickened and failed to send out roots. The plant with the endophyte did not sicken and has flourishing roots. Endophytes can deliver other benefits too, like resistance to environmental stress. For this experiment, Narisawa-san used sweet sorghum. This tropical crop is a source of biofuel, but it doesn't grow well if air temperature is low during sprouting. Narisawa-san tried raising sweet sorghum in conditions replicating early spring in his part of Japan. He tested 115 different strains of endophyte to determine which best supports sweet sorghum growth. In spring 2010, he planted seeds mixed with the best endophyte. They will be replanted in fields for further experiments. He expects to harvest in the summer.
What exactly is the mechanism by which endophytes prevent disease or increase resistance to cold?
Well, it's not a direct mechanism. Endophytes do not directly fight off pathogens. But by establishing themselves in the plant, endophytes elicit the plant's naturally existing resistance to pathogens, cold, or other adverse conditions. For example, looking at pathogen resistance, if there is an endophyte present when the pathogens invade, this causes the plant to switch on its natural resistance. Normally, the pathogens can come rushing in faster than the plant's defense mechanisms can react. But with the endophyte already established, the defense mechanisms are switched on, and the pathogen is unable to invade.
Have the genes for that mechanism been found?
Yes, a number of such genes have been found. But in terms of the overall network, much remains unclear.
You say that much remains unknown. So what do you hope to pursue as the theme of your future research?
As a student, I learned in textbooks that microbes strive to become more powerful than others in order to consume more food. In other words, microbes become elite because they want to eat food. The prime example of this is pathogens. But as we advance with this research, we are learning that's not the case in fact. To a greater extent than we imagine, as we've seen, microbes actually communicate with each other and live cooperatively. In the future, we as researchers need to learn from the microbial world. In our research, we need to cooperate across disciplines in order to open up new frontiers of discovery. That's my research agenda.
Kurosaki-san, your thoughts?
That was fascinating. It's as though I, as a single organism, can only exist through a complex system of symbiosis with all kinds of microbes and other little tiny organisms that we can't even see with our eyes. We learned so many things about the soil ecosystem on today's program. But of all those things, that was what left the most powerful impression on me.
That's all for Science Zero.
See you next time.